Okay, so in the next section, video on online lecture on antibodies, we're going to look at a couple of concepts that you need to know about in understanding antibodies and how antibodies are potentially prepared and the types of antibodies that you will come across. We've seen a little bit of that already by looking at the different classes. There's several different classes of antibodies, and we're going to get into a couple different ways that antibodies are classified, and then also a bit of a description of those antibodies and what they do. Now, the first thing I want to talk about is, or what are referred to as polyclonal antibodies. Now, antibodies can be in polyclonal and monoclonal antibodies, and this has to do with um, the way how we prepare antibodies and the kind of antibodies that you isolate, basically, when you isolate antibodies from an animal, human being, or whatever it is, usually a horse or a sheep, antibodies or a mouse. Antibodies can be used therapeutically and they can be isolated from uh, horses, sheep, mice, humans, and used therapeutically. Now, one of the concepts are what are referred to as polyclonal antibodies. And polyclonal antibodies are a mixture of antibodies which recognize several antigen determinants on one protein. I kind of discussed this in class where there was a question about the antigenic determinants. So these would be polyclonal antibodies is when I have antibodies in a test tube, and the antibodies will all bind to one antigen, but the individual antibodies may bind to different antigen determinants. So if they have different antigenic determinants, say A, B, C, D, polyclonal antibodies will bind different antigen determinants A, B, C, D on the same antigen. Okay, so they can bind those. Um, the antibodies you have an antibody to each one of those. Monoclonal antibodies contain only one antibody, which will recognize only one antigen determinant on an antigen. And so this would be if we just had one of those antigen determinants, we had that antibody, and we're able to clone that out. So if you have an antigenic determinant and it's just one, for example, just A, etc., monoclonal antibodies would bind only one antigen determinant on the same antigen. So this means if I had in a test tube a monoclonal antibody to antigen X, all the antibodies in there would only bind to the antigen determined A on that antigen X. So this is just a sort of a figure to get this a little bit may, may help in getting this idea across. So if we have an antigen in each one of um, these little geometric figures here, the ball, the pentagon, square. Each one of these is an antigen, a determinant on the antigen. And we're going to label those A, B, C, D, E, and F. Okay, so these are all, this is the antigen is the whole thing, and each one of these things is an antigenic determinant. So if I were to have an antibody to the antigen, I'd have one that would bind to A, have one that could bind to antigenic determinant B, one that could bind to the C, one that could bind to D, one that could bind to E, and one that could bind to F. And so all these antibodies bind to the antigen, but they bind to different antigen determinants on that antigen. So if I have a monoclonal antibody, then a monoclonal antibody would be only antigens. Would be only antigens that would bind to B, for example. So it'd have only antibodies in a test tube that would bind to B. And so they would be able to bind to this antigen, but every antibody in that test tube would be a clone of this one. Does it make sense? It's a clone. They're all copies of the same one. So if I had 1,000,000 antibodies in that test tube, all 1,000,000 antibodies in the test tube would be exactly the same. Now, if I have a polyclonal antibody mixture, what I would have is antibodies in which, if I had 1,000,000 antibodies in there, and 1,000,000 antibodies, and each one of those, and there was an equal percentage of each one of those antibodies that could bind each antigen to determine, then 1/6 of those antibodies would bind to these potential antigen determinant. 1/6 would bind to this one, 1/6 would bind to this one, 1/6 would bind to this, etc. And so that would be a mixture, and so they would be able to bind to a test tube of those, referred to as polyclonal antibodies, would be able to bind to multiple antigenic determinants on an antigen. Now, the reasons that you might want a monoclonal antibody and the reasons that you might want a polyclonal antibody, we're going to look at an example a bit later when we look at antibodies that are used as anti-tumor agents, and those are going to be monoclonal antibodies. Later on, we will look at vaccines, referred to as passive immunization, and in most cases, those are polyclonal antibodies. And so, so there are reasons and justifications for wanting one or the other when you're thinking about using these therapeutically. Suffice it to say that if a person gets immunized, and a mammal gets immunized, or vertebrate gets immunized, if I were to look into your blood after you got immunized, I would find, and I had an antigen, and I isolated those antibodies, those antibodies would be polyclonal. In order to make monoclonal antibodies, you have to go through using biotechnology, technology called hybridoma technology, in order to produce actually an antibody called a monoclonal antibody that would be used potentially therapeutically, or maybe diagnostically. You'll learn more about that, how to do that, and the implications for that, not in this class, but you'll learn about it when in the biotechnology class later on. I believe later on, either in the following year. But I want you to be aware of what the difference is between monoclonal and polyclonal antibodies, because you are going to see this term off and on when you refer to them. Now, if we look at the different classes of antibodies, we're going to take a look at the different classes we've already mentioned. The first class we're going to look at are IgGs. These again are the Y-shaped ones, and the crystal structure that I showed you was for an IgG. So they're about 150 kilodaltons in size. Essentially, each chain is the light chain is about 50 Dalton is about 50 kilodaltons, and that's right, is about 50 kilodaltons. Right, it's about 50 kilodaltons, and the heavy chain is about. Sorry about that. The light chain is about 25 kilodaltons, and the heavy chain is about 50. So you combine those together and you get 150. So there are several classes of these. So as I said, there's IgG1, IgG2, IgG3, and IgG4 subclasses, and they're not all equal. IgG1 is by far the greatest in abundance. It has it's about 70%. If I were to look in the plasma and isolated antibodies see there, I'd see 70% IgG1, and everything else would comprise IgG2, 3, and 4, with the next one, IgG2, being about 19%. The subclasses are distinguished by differences in their FC region. So those FC regions have different, they're a bit different in their amino acid sequence. So they have, and as a consequence, that kind of determines a bit as to their differences in what they do and what their potential interactions are with their receptors. And so that subclass, the different subclasses are due to those bit differences in those FC regions, which means there are differences in the heavy chain gamma sequences that define each one of these subclasses. Now, as we're going to say, some of these will bind to FC receptors. We're going to see in a moment how that happens. That will bind to macrophages in phagocytic cells. They will also fix complement. The C prime should change this. This should be complement, and we're going to see about that. They're predominant immunoglobulin in the pool. So these are the, if you look at antibodies in the plasma, it's 70-75% IgGs. And they're the major antibody at second response that has antitoxin activity. So as we'll see later, you can use antibodies therapeutically to remove or eliminate toxins, for example, from bacterial toxins from diphtheria, for example. The next class are IgMs, and they comprise about 10% of the immunoglobulin pool. And these are very different looking in terms of what their final structure is. They consist of five. They're actually in their final. There are five of these immunoglobulin units stitched together, and so each one of those, remember, they're bigger. They have a longer, one more constant region. They have and they have a J, basically as joining chain. And I'll show you right now what that structure is. So if you have, this is the monomeric form. When these are produced and excreted, they actually they assemble into this pentameric IgM form. Is what the final form is. So this is the monomer, and they are assembled into what is referred to as a pentamer form. And this is what you would find in the plasma. As a consequence, an IgM is one of the biggest molecules that you see in the plasma. It's about almost a million Daltons, and it has this pentameric structure because it's so large. It also has, because it's five, right? There's 2 binding sites for each antibody, and there's 5 of these IgMs, so there's 10 binding sites. And you could sort of see, this is an electron micrograph of this, and you can actually see here, you see that that's actually one of the arms. You can see the arms kind of sticking out there, and you could see the arms, so that would be the area that would bind to the antigen. And you can see kind of how flexible they are. 1, 2, 3, 4, 5 of these, and then the joining region. And the reason they're individually, they have the binding site for these antibody at the variable regions might not be the strong, are generally not as strong as for IgG, but because they can bind, because you have 10 binding sites, when they do bind to something and they have 10 potential places that they can bind, they bind very, very tightly, because it's the same principle as if you had if there was a car going past you slowly and you wanted to stop it, and you put your 2 hands on there and you couldn't stop it. But if you have a crowd around there and they grab a hold of the car, they can maybe stop it, because they can grab a hold of it and stop it, because each one of the binding interactions, it's an additive response. The free energy gets added up, is additive. So they're binding. So they bind to the dissociation constants of each of the monomers. It's multiplicative, and so you can and it's additive. The free energy of binding is additive. So therefore, for an antibody for a monoclonal for a IgM, you can have a for its binding site could be weaker than for an Ig with IgG, but it will bind tight. It could have a higher binding affinity in the end due to its avidity effect, the fact that you have many more binding sites that can bind to it than you would have for an IgG. So that's one of the things. It's a potent antibody. Um, it's usually confined to just pool, the to the blood. It's early antibody response. It's very effective at antibacterial antibody, and we're going to see why in a bit. Um, and let's see here, produced and you'll find this on the surface of B cells, and before it's excreted, it's an early response to an infection. In fact, it's rather an ancient response, and so these are really important in the early days of an infection. We'll see more about that later when we talk about the antibody mediated immunity. The next group are IgAs. Now, IgAs comprise about 15 to 20% of an Ig pool that we would be able to isolate. They're the predominant secretory antibody. These antibodies are the ones that get excreted in the sweat into the GI tract, the respiratory tract, urinary tract, and they're very important for protecting against any sort of infections that would go through that transmission route that would it goes through the transmission route through any of those areas. So, for example, they would be important for protecting against urinary tract infections or vaginal infections. Most importantly, we see them in GI infections. So, for example, IgA is really important in protection. It can be for protection against poliovirus or any viruses that norovirus, any viruses that are going to be transmitted through the GI tract, for example. So if you get immunity, if you do have immunity and you have IgA molecule, that immunity for those viruses, you want that antibody, you want IgA-based immunity that's excreted. They have an interesting structure because the monomers are stitched together by a joining region when they're excreted during the excretion process to form dimeric molecules. And so there's two antibodies that get coupled together. Here's an electron microgram, and you can see the two ends here and the two ends here around the staining here. So here's the two variable regions and the two variable regions here, and so they're able to bind 4 binding sites. Okay, so IgM has 10 binding sites. IgA has 2 antibodies, so it's able to bind to 4 antigen determinants simultaneously. The last class is well, it's not the last class, but it's the IgDs. These are rare. They're monomeric. They're relatively abundant on the surface of B lymphocytes. They're really involved in early stages of IgM and to IgG production. This switching is called class switching, and they're not really important in terms of antibodies that are produced and excreted as for the immune response. They're really important they're important, at least we think, in terms of how antibodies are produced within those cells, and they're pretty rare in terms if you see excreted. The last class we're going to introduce are IgEs. Now these have a very small amount in the blood, and that's a good thing because they mediate allergic reactions, and they are the key thing, as we will see when we talk about hypersensitivity reactions. They're the key protein in mediating type 1 hypersensitivity reactions, anaphylaxis, asthma, allergic rhinitis, referred to as hay fever. So these antibodies are involved with that interaction and in the activation of mast cells. We will talk about that later on, but and for now this is just to introduce you to that. They're about this. They're a little bit bigger. Remember, they have a different, a bit longer constant region. About one more, I think one more constant region for heavy chain, and they bind to the surface of basophils and mast cells or typothecal cell. Mast cells, of course, are really important. They line the epithelial airways, the bronchial airways, and they're around in the epithelial cell. They're near their epithelial cells, so they have receptors for this antibody and react with allergens through this antibody. We'll discuss more of that mechanism later in the course. So this is just a summary table of antibodies. You do not have to memorize this, but what I want to point out is just the different characteristics in some. I like this table because it has a lot of nice information on it that I'll just kind of go through here just to show you some of the different characteristics. Now remember the heavy chains up here, the heavy chains that for IgGs, you have 4 different types of heavy chain gamma, 2 different types of heavy chain alpha. There's actually 2 different subtypes, one type of heavy chain for mu, epsilon, one heavy chain delta IgD. Serum levels you can see here. These are serum levels mg per mil. IgG1 dominates, and whole IgGs dominate everything, followed by, if you look at this, IgA and then IgM. This serum half-life is quite a long time. In days, it's almost 3 weeks for IgGs, IgG1, IgG2, and IgG4. And for IgA, it's about a week. IgM also about a week, 5 to 7 days, 6 days. And IgE, it's about 2 to 5 days. This is a complement activation. I'm going to introduce you to that in the next lecture. But just to show you that, IgMs, when you can go back, IgGs are involved in complement activation, and IgAs are not, and IgMs are very involved in complement activation. This is an antibacterial activation. IgGs cross the placenta. Notice this. So these are the ones that provide the fetus with protection, that cross the placenta from the mother. Her paternal antibodies provide the fetus with protection during gestation, and then after birth, those antibodies will be in circulation in the new child, for a period of time. They're also you should also note that in mother's milk, antibodies are also passed through mother's milk and provide protection to the child. And that's one reason. That mother's milk is very good, because there is some of it produced. Some of the antibodies are produced to provide protection to the child through that. In the early life of the child in that way. On B cells, we'll see that the IgMs are on B cells. FC receptors, and they have FC receptors and can undergo phagocytosis. And you can see here IgG1s and IgG3s can do that. They bind to the receptors, as we see, and can do that. You can undergo activation. And IgG1s have the highest ability to do that. And let's see here, ones. IgAs do not, and IgM we're not sure about. Mucosal transport. There is some mucosal transport of IgMs. And IgAs, right? These are particularly IgAs, because remember, these are the ones that are excreted, but IgGs are not. Okay, and the activation of mast cells, only IgEs do this, and we'll see this later when we talk about hypersensitivity reactions. But this is a chart you can kind of go back to, you can look at, and has a lot of the functions in it. So if we look at antibody structure, just to remind you, again, you have the light chain, you have the heavy chain here, this red heavy chain, the light chain, the two arms of the antibody, the variable regions that bind, and then you have the FC domain, this FC region, which is composed of the constant regions from the heavy chain. Now, this part is important because, as I said before, this FC region is important because it can bind to receptors. Now, this is where the use of why antibodies can be so effective in terms of elimination of pathogens or things that they bind to, pathogens or components, or even can attack and kill cancer cells if you can produce an antibody to the cancer cell. And so that mechanism is referred to as antibody dependent cell mediated cytotoxicity, referred to as ADCC. Now, if you have a target tissue, and you have an antigen on that tissue, then for example, a neutrophil, which has antibacterial activity, has FC receptors that will bind to antibodies that are bound to that antigen. And so it will, when it does, it will bind, can release lytic enzymes to degranulate and kill that bacterial tissue cell, and potentially phagocytose it and destroy it. This is true for NK cells as well. NK cells are particularly good at killing cancer cells and tumor cells. They don't do much phagocyte. I don't think much phagocytosis, but they produce lytic enzymes that will degranulate. These are granules that are in the enzymes that get released, and are essentially chemical bombs that get thrown at the cells, full of lytic enzymes. That will destroy the peptide, the proteins, and the lipids, and the sugars to destroy the tumor tissue. Macrophages can do this as well, and if the tissue, if what they're grabbing onto is small enough, they will engulf it. So they can use this antibody FC receptor, if this was a bacteria, to then bring it into or a virus or something, bring it into the macrophage to do phagocytosis. They can also do release of lytic enzymes to destroy the tissue that has for example, if it was a tumor tissue, to destroy the tissue, as these other ones can do. And the last one are eosinophils. You'll see these are in the tissues, and they can go after parasites, for example, and they have receptors that will go after that. You'll see if there's antibodies bound to parasites and things, they'll go after and release lytic enzymes to destroy them. So what you see here is this is an effector mechanism in which the antibodies direct the killing potential of these cells. And this killing potential, if they're small enough, may involve phagocytosis, but in a lot of cases what it does is it involves release of chemically toxic materials, and these generally are enzymes, although in some cases they can be reactive oxygen species or nitric oxide, that will go and kill the tissue, and it will cause apoptosis, programmed cell death in that tissue. So that's how that's how antibodies are connected then to being able to destroy potential pathogen that we've talked about. This is the one mechanism. The next mechanism we'll talk about will be complement, which will be in the next lecture. So NK cells are granulated large lymphocytes. They're 5-10% of the peripheral blood lymphocytes. That's what PBL is, peripheral blood lymphocytes, and they will kill tumors specifically and non-specifically. These, in fact, can be used, and we're using them in the lab. We're engaged in using these in the lab with a collaborator to design immunotherapies using NK cells for target tumors. Neutrophils, these are really important for their antibacterial effect, and they are They adhere to endothelial cells and pass through vascular departments and membranes and into tissues. And so they're very good at going after microorganisms that are in the tissues and also in the blood, but usually in the tissues, blood. They're one of the most important in terms of their antibacterial effects. And when you go under chemotherapy, when a patient is under chemotherapy and that chemotherapy may be toxic to the lymphocytes, it's really important that you follow neutrophils, because if a patient becomes low in their neutrophil count, they can become much more susceptible to into bacterial or viral infection, particularly bacterial infection. Eosinophils, these are like their neutrophils, and they migrate to the blood and to the tissues, and they're important for actually going after parasitic organism defense. Parasites, which usually inhabit tissues, inhabit tissues. So what we're going to do now, we're going to stop now. Here's a set of review questions. And so what I'd like you to do is answer these questions. Stop the video, see if you can answer these questions as a review. Like in the next slide will be the answers to those questions. And we will stop here, and this will be the end of the second part of the lectures on antibodies and antigens.